Hello everyone. Today we'll be solving Cambridge International ASN A-Level Biology Paper 2 Structured Questions, October-November 21, Variant 21. Question number 1. Figure 1.1 is a transmission electron micrograph of the cells from a leaf of a plant. We can see X, Y, Z. The question says name the structures X, Y, Z. So X is nucleus, Y is chloroplast, and Z is large permanent vacuole. State two ways in which the structure of an animal cell differ from a plant cell, such as those shown in figure 1.1. You see, animal cells does not have any chloroplast. There are no large permanent vacuole. There are no cell wall. There are no starch granules present. Animal cells have centrioles present. There is a central nucleus in animal cell. Unlike in the plant cell, the nucleus is in one site. B1. Cell Y in figure 1.1 contains a large starch granule grain. Name the chemical reagent used to test for starch and state the color change that will be seen if starch is present. So the reagent for testing starch will be iodine solution. And the color change that we will be observing is that iodine solution by itself is brown. It will change from brown to blue-black. Starch granules contains amylose and amylopectin. Describe the similarities and differences between the structure of amylose and the structure of amylopectin. Both amylose and amylopectin are polysaccharides. Both are made from alpha glucose. Both are joined by glycosidic bonds. Both contains 1,4 glycosidic bonds. However, amylose are unbranched and amylopectin are branched. Amylopectin contains 1,6 glycosidic bond. Amylose are helical in nature, and amylopectin is more compact molecule. It is branched. Question number 2A. Figure 2.1 is a transmission electron micrograph of a transverse section through a blood capillary that is surrounded by actively respiring cells. All right, we can see labeled fluid R. We can see cell Q. Cell Q is a red blood cell. The fluid R is outside the endothelium layer, so must be tissue fluid. Identify the cell shown in figure 2.1. So the cell shown in figure 2.1 is red blood cell. Fluid R is formed from blood in the blood capillary. State and explain one difference between the composition of fluid R and the composition of the blood in the capillary. So first of all, we're going to talk about the difference. You see, fluid R is tissue fluid. Fluid R is tissue fluid. So, and this is plasma. So fluid R has no large protein or red blood cells. And the explanation is large protein cannot fit through the gaps between the endothelial cells. So red blood cell is too large to fit through the gaps between the endothelial cells. Part B1. The cell surface membrane of respiring cells have roles in regulating the exchange of substances between the cells of the body and blood. Draw a diagram in the space below to show the arrangement of phospholipid molecules in the cell surface membrane. So these are phospholipid molecules. We can see the phosphate head, and we can see the fatty acid tail. Because the fatty acid tails are hydrophobic, so they face each other. Cholesterol is a molecule found in the cell surface membrane. Describe one role of cholesterol in the cell surface membrane. Cholesterol maintains fluidity of the membrane. Nicotine is one of the components of tobacco smoke. Nicotine can cross cell surface membranes and enter the bloodstream. Describe the short-term effect of nicotine on the cardiovascular system. So you see, nicotine can increase heartbeat. It can also increase blood pressure. It can make platelets more stickier. Nicotines can damage the endothelial layer. Nicotine can reduce the flow of blood to extremities. Nicotine is absorbed from blood plasma into the cells of capillary wall. Scientists have suggested that nicotine is transported from the blood plasma into these cells using a type of co-transporter mechanism. Figure 2.2 shows how nicotine may be transported from blood plasma into cells. We can see blood plasma. All right, nicotine is transported against its concentration gradient into cells, and H plus ion is moved down the concentration gradient out of the cell. It's a co-transporter protein. The co-transporter mechanism shown in figure 2.2 is different from the co-transporter mechanism that moves sucrose into the cytoplasm of a companion cell in phloem tissue. Outline the similarities and differences between the co-transport of protein of nicotine and the co-transport of sucrose. Both co-transport have co-transporter protein. Both have H plus ion involved. In both processes, H plus ion moves down the concentration gradient. In both processes, sucrose and nicotine move against the concentration gradient. In nicotine transport, H plus ion and nicotine moves in opposite direction. H plus ion moves from cell wall into cell in sucrose transport. Question number 3. Cholera is an infectious disease caused by bacterial pathogen Vibrio cholera. Describe how V. cholera is transmitted from an infected person to an uninfected person. So feces from infected person can contaminate drinking water. Then if a healthy person drinks the contaminated water, the person will then get infected. V. cholera releases a toxin called choleragen. Choleragen is a protein molecule made up of 6 polypeptide subunits. The figure 3.1 is a ribbon diagram of a molecule of choleragen. So we can see the molecule of choleragen. Okay. This T represents alpha helix. Describe what is shown by the part of the molecule labeled T. So the part labeled T shows secondary structure, alpha helix of a protein, formed by the hydrogen bond between CO and NH of different amino acid. Part C. Vaccination can be used to prevent the spread of cholera. The vaccine used contains part of the collagen molecule and also contains dead cells of V. cholera. Phagocytes play an important role in the immune response to vaccine. Phagocytes contain many lysosomes. Describe the function of lysosome in phagocytes. So basically, lysosomes in phagocytes, they break down and digest pathogen. Lysosome contains hydrolytic enzymes which fuse with the phagocytotic vacuole and digest the bacteria. Question C, part 2. Suggest the advantage of having a vaccine that includes dead cells of V. cholera and part of the collagen molecule. The cells are dead, so cannot cause disease. Only a part of the collagen molecule, so cannot cause any toxic effect. Stimulates the production of antibodies, and this stimulates the production of more than one type of memory cells. D. Part D. The World Health Organization states most cases of cholera can be treated with oral rehydration therapy. To reduce the risk of antibiotic resistance developing, antibiotics are only recommended for the most serious cases. Discuss the consequences of the development of antibiotic resistance in V. cholera. Resistant V. cholera is not killed by antibiotic, so they are unable to treat people with cholera using antibiotics. People staying sick with cholera longer, and this further increases their risk of spreading the cholera. The death rate from cholera then increases. Antibiotic resistance can transfer to other species of bacteria. As a result, other bacterial disease may not be able to treat with antibiotics. Question number 4a. Figure 4.1 is a drawing made by a student of plant cells showing one of the stages of mitosis. Describe the role of spindle during this stage of mitosis. So spindle fibers are attached to centromere. Spindle fibers contract and pull the sister chromatids to opposite poles. Part 2. The chromosomes are composed of DNA. Describe the structure of a monomer of DNA molecule. A DNA molecule consists of deoxyribose, a phosphate, and a nitrogenous base. Part B. Telomeres are located at the ends of each chromosome. Telomeres progressively shorten each time a healthy human cell divides until the cell is no longer able to divide. Stem cells have an enzyme called telomerase that prevents the telomere shortening during the cell cycle. Suggest why telomerase activity is important in stem cells. Stem cells keep dividing throughout their life. Stem cells differentiate into many specialized cells, example, red blood cell and white blood cells and other cells. Stem cells maintain telomeres to prevent the loss of the genes so that they can keep on dividing throughout the life. Part 2. Suggest why tumor cells produce high concentration of telomerase. Tumor cells requires high concentration of telomerase because it allows continuous cell division. Part 3. Scientists have researched the possibility of treating cancer by inhibiting the action of telomerase. One method that has been studied involves inserting small single-stranded DNA into the cancer cells. These sections of DNA have a base sequence that is complementary to a section of the mRNA transcribed from the gene coding for telomerase. Explain how inserting small single-stranded DNA could prevent translation of the mRNA to produce telomerase. So basically, DNA binds to the complementary section of mRNA. As a result, the mRNA cannot bind to the ribosome. The ribosome cannot move along the mRNA. tRNA anticodons cannot form hydrogen bond with mRNA codons. The DNA enters nucleus to bind an mRNA, then becomes too large to leave the nuclear pore, to leave via the nuclear pore. Question 5. A student investigated the effect of air movement on the rate of transpiration by using a potometer. Define the term transpiration. Transpiration is basically the loss of water vapor from the aerial parts of the plant. The potometer used by the student shown in figure 5.1, we can see reservoir of water, screw clip, and leafy shoot that is used in the experiment. And then the air bubble, it travels forward. It travels forward when basically water evaporates. The student recorded the distance the air bubble travel in the apparatus have moved after 10 minutes with no air movement. The rate of water uptake was used as a measure of the rate of transpiration. The apparatus was then reset using the reservoir, and the experiment was repeated a further 2 times. All other variables were standardized during the experiment. Table 5.1 shows the result recorded by the student. So experiment 1. Distance moved by the bubble in 10 minutes / millimeter. So in 10 minutes, we can see in 1, 12.5. In 2 experiment, it moves 12.0, and 3, 11.5. Use the table 5.1 to calculate the mean rate of movement of bubble. So basically, we'll have to add 12.5, 12, and 11.5. 12.5 plus 12 plus 11.5. And then we'll have to divide it by 30 minutes because each experiment was done for 10 minutes. So 1.2 millimeter per minute. The student carried out another experiment using the same plant. In this experiment, a fan was used to blow air across the leaves of the plant. All other variables were standardized. The results showed that the bubble moved further in 10 minutes. The student concluded that air movement increases the rate of transpiration. Explain why the air movement increases the rate of transpiration. Water vapor is moved away from the surface of the leaf as the air blows. This makes the water potential gradient within the leaf and outside the leaf higher. It's more steeper. As a result, higher rate of evaporation into the airspace of the leaf. Next question, part D. Explain how water moves up through a xylem vessel in the stem of the plant in the potometer, shown in figure 5.1. Water moves due to cohesion tension. Hydrogen bonds between water molecules allows the water to move as an unbroken column. Adhesion of water molecule to the cellulose lining allows the water to move as an unbroken column. Evaporation of water results in lower water potential at the top of the xylem vessel. So water moves down the water potential gradient against the gravity up through the xylem vessel. Question 6. In figure 6.1 is a diagram of the section through the heart showing 2 stages of cardiac cycle, A and B. So we can see stage A, which is the atrial systole. Okay, stage A actually is atrial and ventricular diastole, and stage B is atrial systole. Name stage B in figure 6.1. State one piece of evidence from the diagram that supports your answer. So stage B is actually atrial systole. We can see the atria pumping blood into the ventricle. And we can also see the atrioventricular valves open. Draw a label line in figure 6.1 to identify one semilunar valve, label S. So we can see this is a semilunar valve, and that should be labeled S. Last question. Describe the role of semilunar valve in cardiac cycle. Semilunar valve prevents the backflow of blood from the pulmonary artery to the right ventricle. Guys, thank you for joining our class today. If you like the video, please comment in the video and subscribe to our channel. Share it with your friends. It may help someone, those who are giving their exam, and hope to see you next time. Bye bye.